Well, let's watch the news, and they just got done with, I think, a $3 million space project up on the International Space Station. And the experiment was puts a microbial experiment on the outside of the space station to see if microbes can survive 300 degrees below zero. And it was put up there by the stellar evolutionists. They're still absolutely convinced that meteors are the ones that brought how human life and everything life on the Earth came to be on the planet Earth, which isn't true. What they discovered is not even microbes can survive 300 degrees below zero. So now we have to put our thinking caps on again and think in terms of how life on the planet Earth got created. And that the Earth has an extraterrestrial origin. So, now we get back into astrobiology. Because you can't go into astrobiology without design. And so we have to get into the nitty-gritties of DNA and RNA and all the elements of life. And we also have to figure out what kind of spacecraft could be put together out there that can survive a trip with all the gamma ray bombardment and all that. It's out there in interstellar space, so that we could actually survive out there. Because the nearest stars to our planet, and that may be where they're at, is between 5 and 11 light years away from our planet Earth. So that's even going light speed. So we have to think in terms that even if we're able to go light speed, we're going to spend at least 5 to 11 years in space on the trip out there, and on the trip back another 5 to 11 years. So that's still a lot of time in space, even getting up to light speed. So you have to start thinking in terms of astrobiology. So they start studying putting stuff up there, stuff like that, and they discover even microbes can't survive up there in interstellar space. So now we get back down to some recent studies. Something got completed recently that's a scientific achievement, the Genome project, which basically is contains all the DNA and RNA information sequencing to create human life, to create us. So it's never been done before. They've had parts of it that banks they created all over the world, and they added that to it to help speed up the project. So it's not like they had to start from scratch, but it took them a while. It took them quite a few years. They started back in '99, I think, or '98, I think, and they've been slowly putting that together. So we finally got all the information that we need, that you would have to create human life. So now and if you add that up, take the whole project, all the information, it'd go from New York City all the way to London. That's how much information it takes to create this human body. So it's a lot of information in one single witch's ball. You know, it's the miracle of life. We don't know what goes on during that time. That's why it's continued. It's important to keep studying what's going on and the layering and stuff, and actually get sensors down there to see what's going on during that time period where the witch's ball becomes human life. So, but we know that DNA and RNA, DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And DNA and RNA are polymeric molecules made up of linear unbranched chains of monomeric substances called nucleotides. Each nucleotide has 3 parts: sugar, phosphate, and a base. And DNA is a sugar, which is cytosine, guanine, and thymine. Nucleotides are linked by polyester bonds to form a DNA polymer or a polynucleotide, which might be several million nucleotides in length. So it's the nuclear genome is about 3 million base pairs of DNA. So that's how much storage information it's got. So it tells you that it takes a lot of information to create this. So where does all this information from the human genome project going? Well, it's going into a supermax computer because it takes a lot of information, and I think the federal government has that computer. It's one of the supermaxes out there. But it takes a lot of speed and information, process all that information that goes into one federal supermax computer. So, earlier in one of my lectures, I said that ET, intelligent design, how they create life forms, is they can actually do it by computer simulation on a computer. It's a quantum computer. It's not a computer we will have for another 200 years probably, unless it's delivered to us. But it's a very advanced form of computer that combines quantum physics and fields and fusion science and stuff that we don't have yet. But it's getting there, and this is kind of a good time to be around, because some of that stuff we're getting answers on, and so. And there are also going to be quite a few cures that they'll be able to find, because they've got they can look up the genetics of where they think some of these illnesses are coming from, or cancer or whatever. So it's a good time to be around, because we're actually getting some good information. But okay, we got the biologists divide the living world into two types of organisms. Eukaryotes, which is what we are, which whose cells contain membrane-bound compartments, including nucleus and organelles, such as mitochondria. In the case of plant cells, chloroplasts. Eukaryotes includes animals, plants, fungi, and protozoa. So everything that's complex is part of the eukaryotes, which are also bigger molecules too. When you need more advanced life forms, you have to have bigger molecules, which is what the prokaryotes don't have. They're bacteria and very simple life, but the prokaryotes are also the oldest forms of life on the planet. Now, you go back 5 million years ago, and you're terraforming planets, and you're trying to get the oceans going and stuff, we're thinking in terms of green algae on rocks and stuff. We're thinking in terms of, I did a lecture on it, the stuff that floats around in the ocean, can't think of it right now. But anyways, that's what they've been sending them out there to do plankton. Plankton's also 5 million years old. There's very simple organisms all throughout the ocean. Whales feed off them. There's filtering plants out there that feed off them, but they're the oldest, also very old forms of life. So you're talking 5 million years ago. So, but it's very simple life, and what's going on nowadays in DNA, well, they're able to actually edit some DNA, and they're able to modify bacteria with it. So, and they're also doing samples. I think recently they were sending was it Greg Ventnor, I think, was going out on a sailboat and taking samples of the plankton and stuff and bringing it in, and they're going to run the genome on that. So why do you do that? Well, because it's the basics of life on the planet Earth. So if you're terraforming another planet, you have to know what makes that up, and you have to know how to actually edit and get what you want. So we're actually talking design again, and they are able to do that to a certain limit on bacteria and stuff. Bacteria are very simple compared to what we're composed of. and it's archaea, which are less well studied, have been found in extreme environments. Archaea are also another genome, as archaea are found in hot springs, brine pools, and anaerobic lake bottoms. So that's another very old, the archaea around 5 billion years ago too. That's from the basic forms of life. They found it in springs, they've gone down to the bottom of the ocean, they found them down there. So it's very harsh environments, but that can actually support life. So those are necessary too. So if you're going to another world, you're going to terraform planets, you have to be able to distribute that, and you have to be able to get the biosphere going. So we already got information, and we put our think tanks together that we need to understand that. So, because it's important at least for the basics of planet terraforming. So anyways, you get down to the basics of terraforming, and you have to know how that works. So okay, here's the genome size of the prokaryotes are 0.58, 4.64, and 3.0. The fungi are 12.1, the eukaryotes. Aspergillus is 25.4, and protozoa is about 190. Invertebrates are 100, and you get down to silkworms at 490 and stuff like that. Vertebrates about 400. So they're starting to break those genomes down into how to classify them. So that's all involves information. You can go to websites if they're doing genome research on bacteria, archaea, and eukaryotes, and Homo sapiens are at 3,000, and you can go to their websites if you go get this book here. But it's all good information. One of the most popular means of genome sequencing that they use is the shotgun method, which is a standard approach for sequencing small parakaryote genomes, but the required data analysis becomes disproportionately more complex as the number of fragments increase, or a fraction number of possibly overlaps is given by 2n minus 2n. So there's problems with the shotgun method because you can get overlaps and stuff that could actually screw up your data. So that is one of the problems that they had with the shotgun method, but it is a standard method of doing that. There's two types of genetic maps that they use. Genetic mapping is based on the use of genetic technique to construct maps showing the position of genes and other sequences featuring on a genome. On the case of humans, examination of family history, pedigree, genetic mapping is described in this chapter. Physical mapping is using molecular biology techniques to examine DNA molecules directly in order to construct maps showing the position of sequence features, including genes. Physical mapping is prescribed in the chapter. So that's how they did it in the past, but they got the human genome, they've got those in a data bank and stuff, so they got more good information to begin with. So that's that's what they've done in the past. So, but here's the interesting thing about design and stuff, is our pattern in breaking down the DNA and RNA to sample pieces and stuff that they can actually take information off, usually involves two chemicals to break down RNA and DNA, take the sample, put it on a slide, and sometimes it requires electrolysis with electricity or some kind of chemical reaction or something to get what they want. But it takes these steps, and they have to take the process. It's not something that's occurring in nature. It's something that they have to actually do in a laboratory. So here again, it shows you that the design of human life and everything else out there has a process, and it's all based in information. Yeah, I think recently, Michelle Kaku said that advanced civilizations are based on how they use energy. Well, energy is also part of that process of breaking down DNA and RNA into something they can utilize to create with. Bacteria is very simple, but it's something they can actually edit and stuff and understand over RNA, DNA, which is a lot more complex, and that it just has a nucleotide. And there's three methods of changing bacteria and creating new bacteria: conjunction, transduction, and transformation. So it's conjunction is when two bacteria come into physical contact, the one bacteria transfers DNA to the second bacteria. The transferred DNA can be a copy of cell, as possibly all the donor cells, chromosomes, or a segment of chromosomes, DNA, integrated to a plasmid. The latter is called transfer. Transduction involves transfer of a small segment of DNA up to 50 kilobits from a donor to a recipient via the bacterial phage. Transformation is whereby the recipient cell takes up from its environment a fragment of DNA rarely longer than 50 kilobytes released from a donor cell. So there's only three methods that they use to make changes in that. And so it's not really, it's something they can actually work with. Genetic maps, as I told you, that what ET uses and what we have is two different things. Genetic maps can be very limited resolution. It's not like you can go sit down in front of a computer and get a visualized image when you sequence this DNA in that way, what kind of life form you're going to actually create. But advanced science and something that extraterrestrials use out there, they do have that. There's no question mark as to what they're going to create, and it's not like Dr. Moreau, that movie, I saw the two Dr. Moreau movies, that is not advanced genetical engineering, it's using natural selection, which doesn't work. You can edit colors and stuff with natural selection, but you can't change the concept of human life or anything without advanced genetic reprogramming RNA and DNA. It's impossible. So here again, you get into restriction mapping and stuff, and how they cut up DNA and RNA and get these sequences and stuff. And it's a good example of the gel electrophoresis. Where they take a gel and put the DNA and RNA into that and expose it to these types of solutions and put it on a slide, and they can get the DNA stretched out to where they can actually examine the sequencing and stuff. But here again, it takes a process and of information to actually do all that. So it's not something that just, throwing a softball around a ballpark or something, There's another good example of DNA cloning. What the requirements of that are: they got a mixture of the chromosomes in a vial. They drip the vial, hang it up in the air, and they expose it to a laser and a fluorescent detector. It drips down through the laser and fluorescent detector, goes through a charger and two deflecting plates, and the droplets of chromosomes get separated and into two cisterns. But here again, it's a strategy, an example of science and strategy that's been processed over the years. To be able to get what you want, you have to take those different steps. So there's no arguments that design is all included in that. There's no natural selection. It's done in a laboratory, and it's all done by procedure. Let's see. Dioxide nucleotides. I'm not going to get into that. I marked that one, but it's not something that's really important. Let's see. Okay, here's the shotgun method. You take a DNA extract, cut it into various fragments. They put it in a gel electrophoresis, and then they purify the DNA from the gel. And the DNA fragments are about 1.6 kilobytes to 2.0 kilobytes. And they put it on a slide to prepare a clone library. Obtain in sequences of DNA inserts. So they're putting a stretching of DNA containing a sequence, and they enter it in a computer. So, and that's how we got the genome. So it's not too complex of a method, but it's how they got the data for the DNA, for the Genome project. Okay, I'm going to read this whole section here because it is controversial. A lot of people saw Island to Dr. Moreau. That's not advanced genetic engineering. It gave that field the field of genetics a very bad name, even though it's not really a true movie. It's not how that life is created by just random selection and not knowing what they're going to get. But the Human Genome Project has been controversial since its inception. That the large-scale sequencing is getting underway, a number of ethical issues are being raised. One controversy concerns the question of who anyone will own human DNA sequences. To many, the idea of ownership of DNA sequences is a peculiar concept where large sums of money can be made from the information obtained in a human genome, for example, by using gene sequences to direct development of new drugs and therapies against cancer and other diseases. Pharmaceutical companies involved in genome sequences actually want to protect their investments, as they would for any other research enterprise. And currently, the only way of doing this is by patenting the DNA sequences that they discover. Unfortunately, in the past, errors have been made in dealing with the financial issues relating to the research into human biological material. The individual from which the material is obtained not always being a party in the profit sharing, these issues still have to be resolved. The problems relating to the public usage of human genome sequences are even more contentious. A major concern is the possibility that once the sequence is known, individuals whose sequences are considered substandard for whatever reason might be discriminated against. The dangers range from increased insurance premiums for individuals whose sequences include mutations predisposing them to one or more other genetic diseases, to the possibility that racists might attempt to define good and bad sequences and features with dispressedly predictable implications for the individuals unlucky enough to fall into the bad category. Many scientists argue in the support of a Human Genome Diversity Project, which will build up a catalog of sequence variability in different populations and ethnic groups, in expectations that the resulting data will emphasize the unity of the human race by showing that the pattern of genetic variations do not reflect the geographical and political groups that humans have adopted in the last few centuries. The Human Genome Project, especially in the U.S., continues to support research and debate into the ethical, legal, and social issues raised by genome sequencing. It is particularly in great care to be taken to ensure that the genome sequence that results in the project cannot be identified with any single individual. The DNA that is being cloned and sequenced is taken from individuals who have given full consent of their material to be used in their way for whom anonymity can be guaranteed. So, if you saw that there's a movie Hollywood put out called Gattaca, which is a future movie where they have a full, they take DNA test of all, take the genome sequence of all human inhabitants and stuff involuntarily and put it in the data bank and stuff, and you get these eugenic principle theories and philosophical beliefs that came around in the 1930s and stuff that discriminated against people that had supposedly inferior genetics over people that had advanced genetics and stuff, and they created a whole class society around that. And you know, it was one of the Hollywood movies, and that's, you know, that would not be out there as far as there's another, I would say, as far as advanced science goes. Eventually they'll have a cure for most everything out there. Stephen Hawking died recently from Lou Gehrig's disease, and it's a failure of science. He shouldn't have had to spend the rest of his life in a wheelchair, you know. Because we're not Dr. Leonard McCoy. That we don't have the ability to screen that out. But advanced extraterrestrials do have that, and they have the ability to screen out any kind of genetics that is not going to create a perfectly great functional human being. So, you know, it's just a failure of science, and it's a shame that he had to spend the rest of his life in a wheelchair, and being of the mind that he is. But it shows you that we're not extraterrestrials out there. We're not Dr. Leonard McCoy. So, but so there's a lot of good work ahead as far as genetics go and stuff. Well, that ends this lecture.